embryological evidences now under this. Father of embryology is Ernst Haeckel. Father of modern embryology is von Baer. Now Ernst Haeckel has put forth a concept called recapitulation theory or biogenetic law. Now, according to recapitulation theory, ontogeny repeats phylogeny. Ontogeny repeats phylogeny. That is, embryonic stages of a particular animal resemble their adult ancestors. The ancestral characters are called phylogenetic characters. The embryological characters are called ontogenic characters. So, during ontogeny of an higher animal, various ancestral characters make their appearance. The embryos at various stages resemble their adult ancestors. And this concept is also called recapitulation because various embryonic stages during their development recapitulate characters of their ancestors. Now Haeckel has given example to illustrate this. Tadpole larva of frog. Tadpole larva of frog resembles fish in most of the features because fish is the immediate ancestor of frog. Caterpillar of butterfly. Butterfly insect has a larva called caterpillar. The caterpillar looks like a worm resembling the characters of the immediate ancestor of arthropoda, that is Annelida. That is called recapitulation theory or biogenetic law or ontogeny repeats phylogeny. However, this ontogeny repeats phylogeny of Haeckel was contradicted by von Baer. According to von Baer, ontogeny repeats ontogeny but not phylogeny. That is, during the embryogeny of higher animal, various embryonic stages look like embryonic stages of their ancestors or exhibit some important characters of their ancestors instead of resembling adult ancestors. Instead of resembling the adult ancestors. Like if you take the embryogeny of any animal, development starts with a zygote. Zygote transforms into morula, morula into blastula, blastula into gastrula, gastrula into embryo, adult, early embryo into an adult embryo. Now it is in the zygote stage, it resembles the unicellular organisms like protozoans. When it is in the morula stage, it resembles the characters of the embryo, exhibits the characters of colonial protozoans. When it is in the blastula stage, it may exhibit the characters of cnidarians. When it is in the early gastrula stage, maybe characters of the platyhelminthes. Advanced gastrula stage may be characters of a higher diploblastic animal. Then embryos in the early stages, all of them look alike. Embryos of all the animals look alike. As they start developing, as they start growing and developing, they start diversifying themselves from all the animals. In the early stages, they look alike. As they start developing, they start differing from each other and start acquiring the characters of their parent animals. So this was the concept given by von Baer to contradict Haeckel's ontogeny repeats phylogeny. Serological tests. Serological tests, that is blood serum tests, were conducted by H.F. Nuttall. Serological tests were conducted by H.F. Nuttall. He prepared human antiserum and mixed this human antiserum with the blood of chimpanzee, with the blood of a dog, with the blood of a monkey, and so on and so forth. When the antiserum of human being was mixed with that of chimpanzee, there was 90% precipitate, indicating the close relationship between human being and chimpanzee. When it was mixed with that of monkey, there was only 80% precipitate. When it was mixed with that of dog, there was only 40% precipitate. When it was mixed with that of some other distant related animal, there was no PPT at all. Now on the basis of the amount of PPT formed here now, he established the ancestral relationship between different groups of animals here now. If more PPT is formed, it indicates that they are more closely related. If less amount of PPT is formed, indicate they are distantly related. If there is no PPT here now, it indicates that there is no ancestral relationship between those two groups of animals. So these tests were conducted by H.F. Nuttall and they are referred to as serological tests. Now biochemical recapitulation This concept of biochemical recapitulation was put forth by Needham. Now, according to him, during the development here now, if you study various excreted products of a particular animal here now, you can establish the ancestry or their affinity with their lower animals. For instance, during chick embryo development here now, which lasts for about 21 days, first 4 days, the chick embryo eliminates ammonia as a principal nitrogenous waste here now, indicating its relationship with fish. Okay, indicating its relationship with fish and certain aquatic amphibians like Urodila and Apoda and all. Now from 5th day to 11th day, it eliminates urea, indicating its relationship with adult amphibians like frogs and all, which also eliminate urea here now. Then from 11th day onwards, okay, 11th day onwards, it starts eliminating uric acid, indicating its relationship with reptiles. So adult birds also eliminate uric acid. So from 5th day on 11th day onwards, they start eliminating uric acid here now. That indicates the ancestral relationship between reptilia and aves. So this concept is referred to as biochemical recapitulation and it was put forth by Needham. Lamarckism. Now this concept of Lamarckism was put forth by Jean-Baptiste Lamarck. He was a French evolutionist. Now he has written a book called Philosophie Zoologique. Book called Philosophy Zoologique, and in this Philosophy Zoologique he has mentioned a certain postulates. The postulates of his theory are: number 1, influence of environment, number 2, formation of organs as a result of need or want. Number 3, use and disuse theory. Number 4, inheritance of acquired characters. Inheritance of acquired characters. Among these, the widely accepted concept is the widely popularized concept, I would say, instead of accepting, widely popularized concept is inheritance of acquired characters. It is also called Lamarckian doctrine. Now he has given several examples to illustrate all the 4 postulates of his theory here now. The classical example for Lamarckism is the elongation of neck in giraffe. Elongation of neck in giraffe. This is a classical example put forth by Lamarck to illustrate all his 4 examples. The another one is loss of limbs in snakes. Next one is loss of wings in kiwi. He has given several examples to illustrate all the 4 postulates or the principles mentioned in his treatise. However, his concept was contradicted by several scientists here now. A death blow to his concept was given by August Weismann. August Weismann contradicted Lamarckian hypothesis by conducting experiments on certain rats by cutting their tail for 21 consecutive generations and subsequently crossing their tailless rats and he would see once again tailless rats giving birth to tail rats, And this experiment is also called decardialization experiment. Decardialization experiment, conducted by August Weismann to contradict Lamarckism, However, some of the scientists, supported Lamarckism, and they are referred to as neo-Lamarckists. The neo-Lamarckists are Packard, Spencer, Cope, Osborne, Kammerer, Sumner. Packard, Spencer, Cope, Osborne, Kammerer, Sumner are some of the neo-Lamarckists who supported Lamarckism by conducting experiments on various organisms. Among these, the Kammerer conducted experiments on a blind salamander, a cave blind salamander, that is Urodil, lives in caves, He conducted experiments on that animal to disprove August Weismann's hypothesis and support Lamarckian hypothesis, He made that particular Proteus anguiens, that is cave blind salamander, live in sunlight, When it was forced to live in sunlight, over a period of time it has developed pigmented skin and functional eyes. Then when such a modified animal was crossed with another identical animal, they gave birth to the anguiens with similar features. And that's how the Lamarckian hypothesis was supported by Kammerer. Now I shall give you certain important concepts of Darwinism. Charles Darwin, he was born in England. He travelled in a ship called HMS Beagle along with Charles Lyell and several scientists. He visited Galapagos Islands and conducted experiments on finches, birds called finches, which belong to the genus Geospiza. Then Darwin was inspired by three theories put forth by various scientists. Number one, principles of population put forth by A.R. Malthus, that is population increases always in geometric ratio, whereas food and shelter increase in arithmetic ratio. So this was the concept which mainly inspired Darwin in conceiving the idea of natural selection. The next one is principles of geology put forth by Charles Lyell, which says that gradualism repeats uniformitarianism. Gradualism repeats uniformitarianism. Then the third book which inspired Darwin, tendency of varieties to depart from original types. The tendency of varieties to depart from original types, that is varieties, subspecies or varieties, they start differing from each other and differ from the ancestors, as they start acquiring various morphological changes, Now this concept was put forth by Alfred Russel Wallace. So these three theories, inspired Darwin in conceiving the idea of natural selection. Now on the basis of this, he has written a book called Origin of Species. In that he has mentioned about various postulates. The postulates of Darwinism are: number one, overproduction or prodigality where according to him every animal has got immense reproductive potentialities. Like an elephant, it remains alive for 100 years it attains maturity at 30th year, and after 30th year if it starts reproducing, a female elephant can give birth to 6 young ones The 6 young ones can in turn transform into 19 millions of elephants by 800th generation. They can transform into, or they can give rise to 19 millions of elephants by 800th generation. Whether every animal has got extensive reproductive potentialities, which comes under overproduction or prodigality. Number 2, constancy of population. Constancy of population. Number 3, variations. Number 4, natural selection. Number 5, origin of species. These are the 4 postulates of Darwinism here. Number 1, natural overproduction or prodigality. Number 2, constancy of population. Number 3, variations. Number 4, natural selection. And number 5, origin of species. Now Darwin has taken a best example here to illustrate his concept, that is industrial melanism. Industrial melanism, which is peppered moth. There are 2 kinds of peppered moths here Biston, Betullaria, Carbonaria. This is a black peppered moth. Biston, Betullaria, typhi. This is a grey colored peppered moth. Now in the Manchester region of England first there were grey colored peppered moths Then when the Manchester region, it was polluted thoroughly because of industrialization, there was emergence of black peppered moth. That is, genetic change took place in some of the grey colored peppered moths, so they transformed themselves into black colored So over a period of time there were only black colored peppered moths. The grey colored peppered moths vanished because they were all caught and eaten away by predatory birds. So according to Charles Darwin nature in the form of predatory birds has eliminated all the grey colored moths, because grey colored moths had harmful variation, they had large variation, and they could not they could not undergo changes according to changes in surroundings They could not keep themselves abreast of the changes that occurred in the industrial region of Manchester. That's why all of them were caught and eaten away by predatory birds Only black colored peppered moths flourished and prospered because they could easily mimic with the surroundings and they remained non-recognizable on black colored tree trunks. However, there were several demerits of Darwinism also. The demerits of Darwinism, if you look into, Darwin has explained only about survival of the fittest, but not arrival of the fittest. He has explained only about survival of the fittest but not arrival of the fittest. According to him, small fluctuating variations gave rise to living organisms If small fluctuating variations give rise to the organism, there should be intermediary forms or transitionary forms for every organism. For many animals, the intermediate or transitionary forms are lacking. It indicates that evolution cannot occur because of small fluctuating variations. Darwin called large variations sports or saltations. He did not give much importance to saltations But according to all the other modern evolutionists like Hugo de Vries, then Huxley, then Simpson, so on and so forth, evolution of new species occurs always by large variations but not by small variations, Darwin did not explain about over specialization of certain organs. Did not explain about over specialization of certain organs. Now Darwinism does not explain about co-adaptation. Does not explain about co-adaptation here. Now Darwinism does not distinguish between somatic variations and germinal variations. Does not distinguish between somatic variations and germinal variations. And Darwinism does not explain about vestigial organs. Does not explain about vestigial organs. Okay, so these are the most important contradictions or demerits of Darwin's theory. Apart from origin of species, Darwin has put forth another two concepts. One is sexual selection. The other one is artificial selection. In natural selection, nature selects the organisms with the desirable characters. In sexual selection, female has the ability to select the males which are with the distinct secondary sexual characters. In artificial selection, human being selects the animals on the basis of their superior phenotypic and genotypic characters, and he tries to propagate them by crossing them by different methods. Modern or synthetic theory of evolution, it was put forth by Haeckel, Huxley and Simpson. They supported Darwinism and they took some more characteristics into consideration, some more concepts into consideration, and thereby put forth a hypothesis called modern or synthetic theory of evolution. Now, theory of mutations was put forth by Hugo de Vries. De Vries conducted experiments on a plant called Enothera lamarckiana. Enothera lamarckiana, which is also called evening primrose. Also called evening primrose. When he cultivated these plants in his garden at Amsterdam, apart from the parent variety, he found certain new varieties where he called them mutant varieties, and he named them Enothera zygas, taller than parent plant, Enothera nanella, shorter than parent plant, Enothera brevistylis, with stigma shorter than that of the parent plant, Enothera livifolia, with more number of leaves than that of parent plant. So apart from the parent plant or wild variety, he found all these new varieties. He called the new varieties mutant varieties, and on the basis of this experiment, he has put forth certain postulates. According to him, sudden large heritable variations are called mutations. Darwin called sudden large variations saltations or sports. Bateson called them discontinuous variations, whereas Hugo de Vries called them mutations. So sudden large heritable changes can be called mutations. Number two, always mutant varieties are full-fledged. They don't exist in intermediary form or transitionary form. Then number three, the mutations are not cumulative. They are not cumulative, that is, they are not go on piling up from generation to generation. All of a sudden they crop up at any time. Number four, the animals with mutations are subjected to natural selection. Number five, mutations occur during gametogenesis and at the time of fertilization. Next one, mutations may be useful or harmful. Next one is mutations are reversible. That is, an organism before undergoing mutation is called wild type. When it undergoes mutation, it is referred to as mutant type. Sometimes a mutant type may give up the mutant characters and become normal characters. That he has put forth several postulates on the basis of this experiment, which is referred to as mutation theory. Now, according to the synthetic theory of evolution, the natural selection is divided into three types. Number one, stabilizing selection. It is also called normalizing selection. It is also called centripetal selection. Now in stabilizing selection here now, it always this is a kind of natural selection which operates on a population which lives under congenial environmental conditions. When a population lives in a particular area where the environmental conditions are optimal or congenial here now, all the animals present in that particular habitat exhibit more or less similar morphological features here now. Among those animals, if one or two animals exhibit specialized characters or mutant characters or unique characters, such animals are not allowed to remain along with other animals. They are discarded from that particular population, and thereby uniformity is once again established among those animals. So since all the animals with mutant characters are discarded from time to time, and once again normalcy is brought about among the animals now by natural selection, this kind of selection is called stabilizing selection or normalizing selection or centripetal selection. In centripetal selection, animals from both the extreme ends of the phenotypic distribution are eliminated. The extremely high phenotypic and extremely low phenotypic animals, both of them are discarded, and thereby uniformity is maintained among the organisms. Now an example given for this is in England here now, the newborns with more than 8 pounds of weight, newborns with less than 8 pounds of weight, there was a huge mortality among such newborns now. Mostly the newborns with 8 pounds were alive, and only such newborns were seen in most of the parts of England here now. The next you have another one called directional selection here now. Directional selection influences the population which keeps on undergoing changes from one end of the evolution to the other end. A population, when it lives in a particular habitat, when the habitat undergoes changes in a particular direction, in the same direction even the animals of a particular population also undergo changes. The animals which do not undergo changes in a particular direction are discarded. The animals which undergo changes in a particular direction are allowed to perpetuate and propagate, and only such animals are allowed to survive in that particular habitat here now. Now directional selection, once the animals acquire certain changes now, they are further influenced by stabilizing selection. So there will be first influence of directional selection, followed by there will be influence of stabilizing selection. The best example given for this is mosquitoes developing resistance. Mosquitoes developing resistance against DDT, elongation of neck in giraffe. They are the two examples for directional selection. The next you have third one, disruptive selection. Disruptive selection or diversified selection, or it is also called centrifugal selection. Now in disruptive selection here now, in a particular habitat is divided into several microhabitats. There are different groups of animals belonging to a particular species living in microhabitats here now, exhibiting diversified morphologic features. Every group of animals has equal chance of survival because the selection itself is diversified. Selection, instead of promoting only one type of characters here now, it promotes the animals with diversified characteristic features. That's why in the microhabitats of a particular large habitat here now, animals with diversified characteristic features remain alive, perpetuate and propagate themselves. And in case of the diversified selection, if mating between two microhabitats is arrested here now, they may give rise to subspecies because of piling up of newly formed genes here now. They may give rise to subspecies. Subspecies over a period of time may give rise to new species here now. The best example given for this is sunflower can grow in dry area as well as in wet area here now. There are some sunflower plants which grow in dry area. There are certain varieties of sunflower which grow even in wet area. So these are the three types of selections according to modern or synthetic theory of evolution. Hardy-Weinberg law. This Hardy-Weinberg law, it was put forth by two American scientists, Hardy and Weinberg. According to Hardy and Weinberg, in a large population, gene frequencies and genotype frequencies, allelic frequencies and genotype frequencies remain constant from time to time, provided the population obeys certain rules. What are the rules that are to be followed here now? Number one, population should be large. Number two, there should be panmictic mating. Among the animals, that is, there should be random mating instead of preferential mating. There should be random mating among the animals. Number three, not subjected to any kind of evolutionary force. They are not subjected to any kind of evolutionary force like mutation, natural selection, migration, immigration, emigration, so on and so forth. When they are not subjected to any kind of elemental force of evolution, allelic frequencies remain constant. When the allelic frequencies remain constant, genotype frequencies remain constant. When genotype frequencies remain constant, genetic divergence does not occur. When there is no genetic divergence, evolutionary changes also do not occur. Okay, then the population always obeys Hardy-Weinberg law. Okay, then this is mathematically explained by a principle called p square plus 2pq plus q square is equal to 1, where p square is equal to genotype frequency of homozygous dominant. P square is equal to genotype frequency of homozygous dominant animals. 2pq is equal to genotype frequency of heterozygous dominant animals. Q square is equal to genotype frequency of homozygous recessive animals. That is, if you take a particular population living in a habitat here now, there may be homozygous dominant animals, heterozygous dominant animals, homozygous recessive animals. So genotype frequency of homozygous dominant is called p square. Genotype frequency of heterozygous dominant is called 2pq. Genotype frequency of homozygous recessive is called q square. So p square plus 2pq plus q square is equal to 1. That is, all these frequencies remain constant. They will be equivalent to 1 when the population obeys Hardy-Weinberg law. Then there is another concept called Sewall-Wright effect or genetic drift here now. Scientist Sewall-Wright, American scientist, has put forth this concept called genetic drift here now. According to Sewall-Wright, in a small population, allelic frequencies, genotype frequencies never remain constant. In a large population, according to Hardy and Weinberg, they remain constant. But according to Sewall-Wright, in a small population, these frequencies never remain constant. They keep on changing, and changes occur at random here now. That is, allelic frequencies, certain allelic frequencies which are advantageous may be added up, or certain allelic frequencies which are disadvantageous may be lost here now. Either loss or a gain is only a probability factor. They don't go by any rule or a regulation here now. So changes occur in organism as a result of a chance, that not by a choice here now. So such changes that occur in a small population as a result of probability can be called Sewall-Wright effect or genetic drift here now. Now, genetic drift or Sewall-Wright effect is studied under two headings here now. One is called founder's effect. The other one is called bottleneck effect. Founder's effect is when a small population which is separated from a large population starts living in a small habitat here now. The small population acquires certain changes due to influence of various environmental factors, as a result of that, allelic frequencies and genotype frequencies of the small population will be entirely different from that of the large parent population. One important example is given for the founders effect here now. In the beginning, in the Polynesia, there was a huge overcrowding here now. In order to overcome overcrowding here now, some of the Polynesians migrated from Polynesia to Pitcairn. They started living in Pitcairn Islands here now. In Pitcairn Islands, when they started living, they were exposed to different types of external environmental conditions. As a result, they have undergone changes here now. Those changes have brought about some radical changes in the gene frequencies and genotype frequencies. As a result, the gene frequencies and genotype frequencies of the newly evolved population of Pitcairn Island resemble the Pitcairn people rather than original Polynesian people. Though they have migrated from Polynesia, they started living in Pitcairn Islands and they were exposed to different environmental conditions. As a result, their allelic frequencies have undergone drastic changes, radical changes, and finally, the allelic frequencies of the newly formed human beings were similar to the Pitcairn Island people rather than the original Polynesians. So the Polynesians were founders, the newly migrated Polynesians were the founders for their successors rather than the original Polynesian population. In the same way, there is another one called bottleneck effect. A large population lives in a particular habitat because of various calamities and catastrophes which are referred to as bottlenecks. A large population is reduced to small population. Large population is reduced to small population, and the small population over a period of time in turn multiplies to form a large population. Gene frequencies and genotype frequencies of these newly formed animals will be similar to that of the small population rather than the original parent population which was there before the bottlenecks. So these two concepts also illustrate Sewall-Wright effect or genetic drift. Gene flow. There will be flow of genes from one population to another population as a result of interbreeding. When the breeding is arrested, flow of genes is also arrested. So the genes with diversified characters go on piling up. As a result, they may give rise to new subspecies followed by species, which is called gene flow. Then as a result of gene flow, there may be reduction in the allelic frequency of one population, increase in the allelic frequencies of another population because of the migration, that is immigration or emigration. Genetic load. Genetic load is existence of certain deleterious genes or harmful genes in a particular population which are harmful on one hand, at the same time useful on the other hand. The best example for genetic load is the presence of HBS, HBA gene in certain African tribes. When they carry those alleles HBS and HBA, they suffer from a disorder called sickle cell trait. We already learnt about this, because of the changes on the 11th chromosome they suffer from certain ailments. Such people are referred to as sickle cell trait people. In such people, whenever the RBCs enter veins, because of low oxygen tension, they become sickle shaped and RBCs get distorted. Hemoglobin solidifies partially. It carries less amount of oxygen. Sickle cell trait people, though they suffer from certain ailments, they turn out to be resistant to malaria. Falciparum, incidence of falciparum is very high in South African countries. But people with this sickle cell trait do not get affected by falciparum. It becomes a blessing in disguise for them. So such existence of deleterious genes which are useful as well as harmful can be called genetic load. Speciation. Formation of a new species from ancestor can be called speciation. It is of several types. First one is phyletic change or anagenesis. And there is evolution of an ancestor into a newly formed organism through a single genetic lineage evolution of an ancestor into a newly formed organism through a single genetic lineage now, where this newly formed organism entirely differs from the ancestor because of the radical changes that have taken place in the course of evolution, can be called phyletic change or anagenesis. Like human evolution is the best example. Human being has evolved from anthropoid in apes. In course of evolution, human being has undergone drastic changes now. As a result, he entirely differs from his ancestor. Cladogenesis, fragmentation of a single lineage to form several lineages in course of evolution, can be called fragmentation. The best example for this, Reptilia, class Reptilia, there is a group called Ornithischia. These ornithischians are herbivore dinosaurs. They gave rise to birds. And there is a group called Theropsida. Theropsidans gave rise to mammals. So ornithischian dinosaurs of Reptilia gave rise to apes. Theropsidan reptiles gave rise to mammals. Whether a single lineage through fragmentation gives rise to 2 to 3 lineages in course of evolution, it can be called cladogenesis. Microevolution. If changes occur in a particular group of organisms in such a way that it leads to formation of a new species or below the level of species, it is called microevolution. Some minute changes, small changes occur. As a result, it becomes inevitable for the taxonomists to create a new species or a subspecies from a particular group of organisms. It is called microevolution. Macroevolution, if changes occur in a large scale which leads to the formation of a taxon above the species level. Leads to the formation of a taxon above the species level, maybe family or an order or a class or suborder, so on and so forth. So above the species level, the changes that occur which lead to the formation of a new taxon above the species level, such changes can be called macroevolution. Allopatric speciation. Allopatric speciation, a large population lives in a particular habitat. The large population is divided into 2 to 3 habitats because of some geographical barrier. Now the 2 to 3 populations, when they are separated from each other they undergo various changes, genetic changes because of influence of external factors. Genetic divergence crops up in some small subpopulations. The genetic divergence leads to reproductive isolation, and reproductive isolation over a period of time leads to formation of a new species. Whether if evolution of a new species takes place because of reproductive isolation, genetic divergence and geographical barrier. First there will be influence of geographical barrier. Geographical barrier leads to genetic divergence. Genetic divergence leads to reproductive isolation. Reproductive isolation leads to new evolution of new species, which is called allopatric speciation. So 2 to 3 subpopulations of a large population over a period of time give rise to new species through geographical barriers, genetic divergence and reproductive isolation. It is called allopatric speciation. Sympatric speciation. In a large population there may be 2 subgroups of a population which live together in the same habitat, but they differ from each other behaviorally and physiologically. Maybe in the large population, 3/4 of the population may be ferocious group, 1/4 of the population is meek and submissive group, where these 2 populations are separated from each other behaviorally and physiologically, and these 2 subpopulations over a period of time, if they give rise to new species through genetic divergence and reproductive isolation, that kind of speciation is called sympatric speciation. So the main crux of sympatric speciation is behavioral differences and physiological differences will give rise to reproductive isolation, and reproductive isolation in turn leads to formation of new species. In allopatric speciation, geographical barrier causes genetic divergence. Genetic divergence leads to reproductive isolation. Reproductive isolation in turn gives rise to new species, which is called allopatric speciation. So these are some of the most important concepts in the evolution lesson here, organic evolution lesson. Now we shall take up certain MCQs on evolution here.